Hello, and here we are with week 4. This week's SSV is about nutrient stoichiometry and nutrient acquisition in carnivorous plants, specifically pitcher plants. So before we dive into the study, it's important let's learn a little about pitcher plants in general. Pitcher plants, like other carnivorous plants, grow in really nutrient-poor environments, and they have adapted by supplementing their nutrient intake by capturing insects and other organisms and digesting them inside the pitchers with enzymes, as well as bacteria. They produce both pitchers and photosynthetic leaves based on the relative nutrient inputs. Now, the species that we're going to be talking about today is called Sarcinia purpurea, and it's distributed all along the East Coast and throughout Canada. So it's a pretty widespread species. Now, the importance of studying nutrients in pitcher plants is because it'll help us understand the relative nutrient uptake in these species, as well as the underlying physiology of a given plant species and what are the relative nutrient needs. So in terms of nitrogen, phosphorus, and potassium, which are 3 of the key macronutrients. It also could help us understand patterns of species distribution, abundance, and population dynamics. So why is this species here? Why is this species there? For pitcher plants in general, they get their nutrients through the pitchers in 2 different ways. One is through captured prey, and once this prey is decomposed, it provides nitrogen, phosphorus, potassium, calcium, and other nutrients. While atmospheric deposition, so just pure kind of dust, if you will, provides primarily forms of nitrogen, so it's either nitrates or ammonia, and that doesn't include the other essential elements. So this potentially could play out in terms of the stoichiometry of the plant. So the researchers decided to investigate what is the effect of prey addition on the morphology and photosynthetic rates of Sarcinia purpurea. So what are the physiological consequences of prey addition, of capturing organisms? Basically, how does this captured prey influence plant-level characteristics? So their hypothesis was that with a response to additional prey, plants would increase in leaf biomass, so the leaves would get bigger. They would upregulate their photosynthetic rate, so they would photosynthesize more because of this nutrient limitation. And they would also increase their nutrient absorption. Now, pitchers are produced every 10 to 14 days, so there's a fair amount of turnover. And they can have an average of maybe 5 leaves annually, so most of the plant is consisting of these pitchers. And these pitchers that last several weeks before decomposing have a very low capture efficiency. So for all of the potential prey that visit a given pitcher, only about 1% of that potential prey that visit the pitcher are actually captured. So there's a very low capture rate. But it could be enough to supplement the nutrient needs. So the way they conducted the study was in a Vermont bog. They selected 200 adult plants that were at least 50 centimeters from one another. So for the sake of argument, we'll assume this grid is 200 plants and it's spaced randomly throughout the bog. And they're randomly assigned one of 8 different treatments. So 0 flies added per week, control 2, 4, 6, 8, all the way to 14 a week. And they were fed frozen house flies throughout the summer. And they were selected at random to avoid selection bias and accommodate the inherent variation amongst plants. Now, throughout the summer, they would randomly select some of the plants from each treatment for pitcher collection, measuring the physical and chemical characteristics of the pitchers back in the lab. Now, the results were that they found when they added flies to these plants, they recognized increases in leaf nitrogen as well as increases in leaf phosphorus. So the addition of flies provided a benefit in terms of nitrogen and phosphorus. However, there was no effect for calcium, potassium, and magnesium, which is interesting considering those are the micronutrients that are most associated with prey addition not necessarily nitrogen deposition. They also found that collectively these prey additions shifted plants from being phosphorus limited under ambient conditions to being nitrogen limited, or nitrogen potassium co-limited when prey was added. And so let me walk you through this graph right here. So right here, these are threshold values that represent limitations for a particular ratio. So this is kind of from previous literature, these are inherent thresholds which determine which nutrient is limiting for a given plant. When the value of the ratio is higher than the threshold, then the plant is limited by the denominator nutrient. So this is true for phosphorus at first. So here you can see phosphorus in this first column had a concentration of, or that ratio in particular was 17, which is higher than the threshold. So that means that the plant is initially limited by phosphorus. However, when the ratio is lower than the threshold, the plant is limited by the numerator nutrient. And so that is nitrogen and potassium for the remainder of the experiment, or for the other treatments, I apologize. So when you added flies, when you added prey to these pitchers, they became more limited by nitrogen and potassium than they were by phosphorus. So, and also, in addition, prey addition had no obvious effects on plant morphology, so no change, non-significant results in terms of leaf biomass. Photosynthetic rate, they did not photosynthesize more with more prey, and there seemed to be a slight change in nutrient resorption, especially for nitrogen and potassium. Now, what does this mean? So the nutrient stoichiometry, so this relationship between different nutrients and their concentrations, reflects the nutrient inputs of both prey and atmospheric sources. So although there was no change in morphology and photosynthesis, it's likely that these extra nutrients were being accumulated and stored for future growth and reproduction. And ultimately, pitchers seem to be phosphorus limited without prey addition. However, when you started adding prey, so they started capturing prey, pitchers became more nitrogen limited. And this could be important in the future when nutrient additions to the system or in this environment might shift. And so that's the end of the study. Just this little blurb at the end. I found this online. There's this really cool video of a pitcher plant in a botanic garden in Great Britain where it actually consumed a bird. So I don't know how it did this, but carnivorous plants are just too cool for school. Feel free to check out the videos online. They're pretty neat. Thanks.